Hello, my name is AdamHales, and I'm an undergraduate student at Utah Valley University in Orem, Utah, the Department of Biology, and I work in a plant breeding and genetics lab. And this project was to look into genetic resistance to a DNA virus in Brassica species. So within the Brassica genus, there's a huge variety of species and plants that are grown, commonly food crops. Broccoli, cauliflower, cabbage, kale are all ones that come to mind right off the bat. But a very diverse genus, and one that was really cool to use for this study because of its diversity. There were so many different species that could be requested from all over the world to help screen for DNA virus resistance in this study. And you can see there that graphic, just the diversity within this genus. So here you can see a picture of the very first plant that was discovered with this virus here in Utah by Dr. Al Mullaney, our virologist, and his lab. They went to this plant and they removed an aphid that was on this plant, and that's how they determined it was a virus disease along with the symptoms of this plant. They removed that aphid, they grew it in culture, and continued to feed it new tissue and its progeny new tissue to develop a colony of symptomatic aphids that transmit this virus throughout time, and that's the same progeny that we've used throughout this experiment. You'll notice there some of the symptoms that we observed in this plant, discoloration in the leaves, leading to dry and brittle leaves of this plant. So as mentioned, this virus is vectored via an aphid, or more specifically the cabbage aphid. You can see there on that photo those aphids on leaf tissue, and they work by infecting the plant with their mouth parts. They'll insert their mouth parts into the leaf tissue, more specifically the phloem, and they'll exchange contents within their body with the plant, and that's how the virus gains entry into the plant. In addition, aphids are a very common vector for plant viruses, and that vector range is increasing with climate change. So as our climate continues to warm and as temperatures increase around the world, these vectors can go into places and to reach regions they've never reached before, thus exposing plants and other organisms to novel pathogens. So all that to say that this problem is certainly going to continue to occur as climate change continues to affect us. The objectives of this study were, first of all, to develop a disease severity rating, or a DSR, disease severity score, a way to quantify how these plants did against the virus. You can see there a photo of a brassica that's done really, really poorly against this virus. You can tell that discolored leaf tissue, very symptomatic response. In addition, another objective was to screen a variety of brassica germplasm to look for regions of DNA or to genetic information that underlie DNA virus resistance in these plants. So 197 accessions were requested from the USDA germplasm system to use in this study. 3 commercial varieties, in addition, were used too, making a total of 200 accessions. You can see here a map of where all those accessions were derived from, where we got them from, those country of origin, as well as their sample count, or the quantity of those that were found in each location. You'll notice it was very common throughout Europe and Asia to have brassica species that were used in this study. And the reason for that is, in Asia especially, there were a lot of cultivars that had been made and bred for virus disease resistance. So it was important for us to accept those and to use those in the study to see if that previously reported virus resistance was applicable to this particular virus. So as mentioned, 200 brassica sections were grown in a greenhouse setting. After 6 weeks of growth, a leaf tissue was harvested and set aside to use for DNA extraction and then sequencing. 1 week after that leaf tissue was harvested, so at 7 weeks total of growing time, these plants were inoculated with the cabbage aphid, and they were allowed 3 weeks for an incubation period. After that 3 weeks, a disease severity rating, or that DSR, was conducted to assess the plant's reaction to the virus. And from there, that evaluation data, as well as the sequencing data, will be combined to use in a genome-wide association study. So in this study, it was important that we use an RCBD design to screen this germplasm, and we used 3 blocks, 3 replicates, 200 sections for each replicate, a total of 600 plants. You can see here this graphic of the greenhouse layout that we used. On the left, the wall of the greenhouse with the vent and large exposure to sunlight, and on the right, the swamp cooler. So the very diverse climate, microclimate within the greenhouse. So that's why that RCBD design was so very important to us. In addition, you'll see a blue line kind of snaking through there. And that's our drip system that we designed for this experiment. We wanted the watering application to be very uniform so that any traits or any reactions to the virus observed in these plants could be traced back to their response to the virus, not some other external factor. So we designed this drip system to make watering very uniform across all the plants and all of the blocks. You can see here a photo, the outline of that drip system, and then a cage over the top of it almost. And that is what we called an aphid dorm in this project. And that kept those aphids inside to infect only those brassica species and not to get out and infect other plants in the greenhouse. In addition, that aphid dorm served as a barrier for pests in that initial growth phase for those plants, so we didn't have any outside pests coming in and infecting those plants with any foreign pathogens. So all these plants were inoculated using a symptomatic leaf tissue culture of those cabbage aphids that were distributed uniformly throughout this block. Aphids have, and these aphids in particular, have what they call a telescoping generations, where in each organism there's several other generations already ready to go. So their growth is very, very rapid and very, very quick. In this study, we aimed to place about one aphid per plant in the initial inoculation, which would then multiply itself with time. In addition, these plants were kept under that fine mesh, that aphid dorm, to keep them from getting out and infecting other plants within the greenhouse, and then again to keep other pests out from getting in and contaminating these plants as well. You'll see here photos of us inoculating these plants. You'll notice us distributing that leaf tissue throughout the blocks. And then the far slide, you can tell that leaf there, and that's where the aphid would reside, and then with time that leaf tissue would dry out, and the aphid would flee from that tissue and move to the growing tissue of the plants that we wanted to screen. So more on this disease severity rating, or the DSR. As mentioned, it was very important to us to develop this system because this had not been done for this virus-host interaction. And so this ranged from a scale of 1 to 10, with 1 being no visible symptoms at all, and 10 being complete plant loss. And this was assessed by leaf discoloration, as noted in those other slides, very common pattern in these infected plants. So we took that leaf discoloration across the whole plant area, more specifically the leaf area, to use for this disease severity rating. Some examples here of what the different disease severity scores look like. We have a 2, a 4, a 5, and a 6. You'll notice there the varying levels of discoloration or lesions in that leaf tissue. And then 6 there, you can notice that extreme discoloration between that vast change in color between the green and the yellow in the plant there. We use that DSR, of course, to screen these plants and quantify their reaction to this virus. So you can see here the different species that were used in this study, everything from Brassica oleracea to the virus-resistant ones. What's notable is the Brassica oleracea had an average DSR of 5.05, so relatively low compared to the other plants. Notable, Brassica gravenae had a very, very low average disease severity rating, but very few accessions were used for this experiment because that was not one that was widely available. In addition, those 3 commercial varieties actually performed very, very well against this virus, and that could be due to their being bred for cultivation and resistance to common garden pests. What's really intriguing about this table, though, is the virus-resistant, or the VR column there. You'll notice that these had a very high average DSR comparatively. It's about 7.1. And so this told us that those virus-resistant cultivars that had previously been bred for were not very effective against this virus, so it really opens up the way for us to ideally create one of those cultivars. Here's a graph showing the distribution of those DSR scores across those different species. You'll notice Brassica oleracea there with about the lowest in most of the sections that were recorded. Again, Brassica gravenae and some other species that were used. Brassica rapa was the one that had a lot of virus-resistant species within it, but it actually did not fare very well. Here's another graphic kind of exploring that virus-resistant response a little more. Brassica oleracea there, it's just a line because there's only one accession that was used, and it was a virus-resistant accession. And then the Brassica rapa were both virus and disease resistance. A more broad disease resistance was noted in the USDA germplasm system, and so we noted that, both of which were pretty high, then again about 7, signifying that those cultivars were not effective against this virus. In addition, I mentioned these accessions were requested from all over the world. We made a graphic here showing the average DSR by origin. And you'll notice here the lowest of which are Bulgaria and Spain, very, very low. And so that's an avenue that we could explore further. And then you'll notice just the wide spread there in origin and disease severity rating. Currently, we're in our second growing season of this experiment using the same experimental design. We've just infected these plants with the symptomatic aphid colony, and we will allow for 3 weeks of an incubation time before we conduct another phenotypic analysis of the response to this virus. After that has been done, we will combine that with the sequencing data that's being processed currently to conduct a genome-wide association study to look for regions of genetic information that underlie virus resistance in this genus. From there, our goal is to develop a variety of this plant that is resistant to this particular virus. So first of all, a big thank you to our funding sources, the UVU Scholarly Activities Committee and the UVU Undergrad Research Scholarly and Creative Activities Grants for funding this project. In addition, I'd like to thank my mentor, Dr. Yuya Liang, for her help and support, my lab mates and Dr. Al Mulaney's virology lab, and specifically, and all others mentioned on this slide. If you'd like to contact me or get in touch with me, feel free to scan that QR code there, and that'll take you, that's a link to my website where you can find my CV, a little bit about me, and all that kind of stuff. So thank you so much for tuning in,